Hey guys, so I wanted to go over this Unit 4 FRQ. Unit 4 was on the cell cycle and looking at how cells communicate with each other. And so we talked a lot about how the regulation of the cell cycle is monitored at different checkpoints, that you could have cycle-independent kinases that are going to allow the cell cycle to progress. You'd also have tumor suppressor genes that could be activated to prevent the cell cycle from occurring, and that at each checkpoint it had to get approved by the cycle-independent kinase before it could enter into the next phase. Remember, the cell cycle is interphase, which consists of G1S and G2 phase, and then it progresses into M phase, which is prophase, metaphase, anaphase, telophase, and cytokinesis. So when we talk about the cell cycle, that happens in your body cells, sometimes called somatic cells, where your original cell is going to be copied, and you're going to create two identical daughter cells from your parent cell. So this would happen in your body when you needed to grow or replace things that have been damaged, or just say you lose a lot of blood, your blood cells could undergo mitosis to replace the lost blood. And every time you replace the cells, you want them to be identical to the original. We have diploid cells that have two sets of their parental chromosomes, and you make another set of diploid cells. Remember, when we have meiosis is the one where we make cells that have half as much genetic material. That happens twice. And if you start with 46 chromosomes in meiosis, you end with 23 chromosomes in your sperm or egg cells, as opposed to mitosis, which is what we're talking about in this prompt, where you start with 46 chromosomes and you make an identical cell that also has 46 chromosomes. So let's look at mitosis and how the cell cycle is regulated. For this, I just pulled up the sketch notes that I gave you guys on this topic. So right here is our diagram. Right here is our diagram about the cell cycle. It says a diploid, that is represented by 2N, is entering the cycle. We have interphase right here, which is made of these three parts, G1, S, and G2. It can also at some point exit the cell cycle into a resting or a non-dividing phase if it is fully differentiated. And so we talked about how brain cells and nerve cells go into this resting phase where they're never planning on dividing again. So first thing the cell is going to do is grow and replicate the organelles so that the new cell has enough organelles. Then it's going to copy its DNA and copy all of its chromosomes. So this is important. I think of it as if I have a worksheet that I need to pass out to my students, I can't just give away my master copy. So the cell can't just give away its master copy of DNA. It needs to go make a copy of it, and then the new cell will have a copy of DNA, and the original cell will retain its DNA. So during the S phase, it stands for synthesis. The chromosomes are duplicated, and that's when we create these sister chromatids. Then in G2, it continues to grow, and then we go into M phase, where it goes prophase. Sometimes they reference prometaphase, metaphase, anaphase, and telophase. And every time we get to a new phase, there's a checkpoint. It's there's a G2 checkpoint to make sure that the chromosomes were replicated properly. And then in metaphase, they make sure that all of the chromosomes are lined up along the equatorial plate properly before they can progress into anaphase. So there's always these different checkpoints that are happening to make sure that the cell cycle is progressing correctly and that you're not spreading mutations or causing problems in the new daughter cell. And then after it goes through cytokinesis and splits into two new cells, you have two identical daughter cells, and you can see they look just like the original. That is 2N, which means a diploid cell, and these are also 2N, which are diploid cells. So we took one cell, and then we split it, and now we have two cells. So remember, the most important part here is that we have this regulation that's happening, and improper regulation can lead to cancer. So cycle-independent kinases are what is going to regulate this internal cell cycle. And then there's also external factors that can influence it, such as density-dependent inhibition, so that when cells are near other cells and they start to get a lot of cells crowding each other, it can turn off the cell cycle. because that's like indications that there could be a tumor. And then this is just like a little to-do list of what happens during mitosis. So the big thing here is understanding what the cell cycle looks like and that it's regulated at every checkpoint to make sure that the cell does not progress and form a mutated cell that could lead to cancer. So let's read our prompt. Okay. The p53 protein is a transcription factor that regulates the cell's progression through the cell cycle after DNA is damaged by UV radiation. The p53 pathway is shown in figure 1. All right, so we look here, we see our DNA damage is here. And after the DNA damage, the arrows are pointing to this protein kinase that's going to interact with p53. And this p53 is going to go inhibit cyclin-dependent kinases. So that's kind of what we're looking at here. It says the DNA is damaged, which activates p53. And then p53 is going to go shut down the cyclin-dependent kinases. Well, remember, the cyclin-dependent kinases are there to help the cell cycle progress, and that it's going to be responsible for the checkpoints that help the cycle continue. So these cyclin-dependent kinases, if they are inhibited, they're going to stop the progression of the cell cycle. Well, if the cell cycle stops, then you're not going to create daughter cells. And if you don't create daughter cells, then you're not going to spread the DNA damage. So what is happening here is this p53 is shutting down the cell cycle so that this DNA damage cannot spread to daughter cells. So a skin cell completes one round of the cell cycle. Describe the products. Well, a skin cell is a body cell, and body cells undergo mitosis. And so what is the product of mitosis? Well, what's what we looked at up here. The product of mitosis is 2 identical daughter cells. So that's all they're looking for here, is that you understand that after a skin cell goes through the cell cycle, the products will be 2 identical daughter cells that are identical to each other, and they're identical to the parent cell. Based on figure 1, explain how p53 regulates the cell cycle in the presence of damaged DNA. So p53 is going to inhibit cyclin-dependent kinases, which would stop the cell cycle so that the damaged DNA could not spread to daughter cells. Draw an X on the template in the space provided for your response to indicate the phase during which the replication of damaged DNA would occur. So if we go down here, this is the place where they want you to put an X. We can go through and say, okay, well, during which phase would we replicate our DNA? So again, hopefully you guys remember that. Or you could just reference here and say, okay, during which of these phases was the DNA replicated or duplicated? And then you can just place an X on that diagram down here. And then lastly, based on figure 1, explain how a mutation to p53 may lead to an increased risk of cancer. Well, p53 is going to shut down the cell cycle by inhibiting the cyclin-dependent kinases that help the cell cycle progress. So you have to think, well, if p53 wasn't working and the cyclin-dependent kinases continued to work and promote the cell to go through the cell cycle, then this DNA would be present in the daughter cells, and we would have damaged DNA going to the daughter cells. And so every time we see damaged DNA, it's going to increase our risk of cancer because we are creating cells that have damaged DNA because we're not properly regulating the cell cycle. So if p53 was not working, it could not shut down the cell cycle. And that way, if it couldn't shut it down, the DNA damage would travel to the daughter cells. So I hope that makes sense. And yes, so just make sure you annotate on here wherever you would put the X, and then just put your answer down here. Hope you guys are doing well, and I will maybe see you soon. I don't know. I will talk to you later. Bye.